Welcome, everyone. Welcome to this year's Gottfried Schmier Memorial Lecture. Before I introduce our esteemed speaker, Dr. Wes Van Voorhees, I wanted to take a little time to remember our friend and colleague, Dr. Schmier, who's pictured here somewhere in Brazil in his second career, which I'll tell you about. So those of you who remember Dr. Schmier will recall that in the first part of his career, after he trained in Austria and studied biochemistry at the University of Vienna, he came here to Seattle to join Earl Davey and others as a postdoc in the late 60s and worked in coagulation, isolating von Willebrand's factor VIII complex, and was recruited by our founder of our department, Paul Strangerd, to come to UW Lab Medicine, where he served as the chief of clinical coagulation service from 1970 until the late 90s. And then, I love this quote from him, because when he retired, he says, that's when the real adventures in my life started. After I retired, I decided to dedicate his years to helping people in the tropics as a volunteer physician. And this is him at the Gorgas, the esteemed Gorgas course in tropical medicine in the late 90s, where you can see that he's the outlier on the upper edge of the age range with the rest of the Gorgas class. And what he did during his retirement is travel to South America quite a bit to work in areas that have various tropical diseases, leishmania, malaria, trypanosomes, et cetera. This is just a few of the places he worked in Peru, in the Amazon with his colleague Sinesio Talhari, who studied leprosy, amongst other things, leishmaniasis. And he worked here in the Hansen's disease clinic until 2016. Some of you will recall that he rode his bike to work, famously injuring himself doing so at an age that most people would have given up biking, and he spoke all these languages. I remember him teaching in the microbiology clinical lab, teaching fellows, teaching residents, et cetera, really kind of giving of himself and being our friend and teacher. So this lecture made possible by Dr. Baird, thank you, Jeff, helps to honor the careers of people who have made great contributions in global health and microbiology, et cetera. Our speaker today is Dr. Wes Van Voorhees, known to many, many people across the infectious disease and microbiology space here at the University of Washington. Dr. Van Voorhees is a professor of medicine in the Division of Allergy and Infectious Diseases, and he has been a part of the fabric of this work in Seattle for 40 years, having gotten his medical license in 1986 in Washington. And his work has focused on the discovery and development of treatments for parasitic and neglected diseases, including in malaria, Cryptosporidiosis, trypanosomiasis. His work has started out in immunology. Some of his very first work was with Ralph Steinman, who discovered dendritic cells and went on to win the Nobel Prize. He's worked in structural biology and translational drug development, also gone to Europe and was inserted into groups at WHO and the Medicines for Malaria venture. And all told, he's published over 350 publications, which is just jaw-dropping. He's been continuously funded by the NIH for decades, and perhaps the most impactful thing is how many people he's trained. Dozens of fellows and graduate students, over 200 undergrads, and interestingly, a fact that I figured out by looking at his CV is that he even trained 2 of our past Gottfried Schmier lecturers. So, Wes, thank you for joining us today, and we look forward to your talk.
Oh, thank you. Wow, you barely recognize yourself when you're introduced so nicely, but thank you, Sean Murphy, and thank you, Susan, for the wonderful invitation to give this talk. I have to say, Gottfried was one of my heroes, or is one of my heroes still. So I looked for a picture on him on the internet, and I could only find this bloody LinkedIn picture where he's sort of scowling a little bit, and I never experienced that. He was always so joyful in his plate round teaching, like you showed, and in carrying out. And he had set up a trip with me to go visit his friend Sinesio when they had this awful weather catastrophe, and I wasn't able to carry it out. But he was always so excited about tropical medicine, and This is a tropical medicine kind of problem I'll talk about today, and I'd really like to dedicate this lecture to Gottfried, or as Sinesio called him, Gottofredo. I want to start out by pointing out that I have a conflict of interest. I am an officer and a founder of Paratherotech LLC, which is a biotech company looking to develop BKIs for animal health. You'll hear all about BKIs today when I'm talking about it for human health. My partner and I really hope that we're going to be able to get some profits in the animal health area to support the development of the drug for humans, which has been a pathway for some antiparasitic drugs that come in through veterinary use and then make it into underserved populations in humans. So the title of the structure is Structure-Guided Drug Development for Cryptosporidiosis, with the goal to save hundreds of thousands of children's lives annually. One other thing I want to talk about before I get to involved slide is a little outline of the talk. I'm going to talk about the problem of Cryptosporidium infection or Cryptosporidiosis, and how it really impacts especially children in low and middle income countries, how the current drug that we have for it is really inadequate to help that population and immunocompromised patients. And then I'll talk to you about the process of structure-guided drug development and how we used it to come up with very potent drugs right from the beginning, but then used it to find alternative drugs that had better and better safety and pharmacokinetic profiles. So hopefully you'll learn something about that drug discovery process, which is a bit iterative and takes a lot of luck. So anyway, Cryptosporidium is a single-celled parasite, a protozoan parasite in the same class of the parasite Sean loves, which is apicomplexa. Sean loves Plasmodium, which is, is the parasite for malaria, also in the group with Toxoplasma. And it's truly a One Health disease, or One Health disease because animals can get Cryptosporidiosis and actually pass it on to humans, or we can pass it back and forth. Calves, for instance, suffer prolonged diarrhea, stunting, and death with co-infections, and stool from infected calves where they pass over the course of their infection, something like 10 to the 13 infectious oocysts can pass into water sources and get into the human cycle. But interestingly, in the developing world where it's so frequent, it's usually human to human that there's transmission is going on, probably from poor sanitation and so on. So these oocysts that can be drank or on food or from person-to-person contact can be ingested, and then they undergo a replicative cycle in the gut. Unlike malaria, they don't require an insect or an intermediate host, if you will, but they undergo a full replicative cycle 2 or 3 times before they get into a sexual cycle in our own guts, and then they form these thick-walled oocysts, which are now infectious to people. And the problem with them is they're not sensitive to chlorination or other chemical other chemical means, but they need to be filtered out. So I'll talk about when filtration breaks down. We have large outbreaks even in the developed world. But they undergo this complex life cycle, and they're right on the edge of epithelial cells, as you can see in this picture up here. Let me see if I can figure out how to point. It's right here along the edge of the epithelial cells in the lumen, so in where the stool is passing, they undergo their full replicative cycle. And so we need to think about that in terms of drug development. So they have a tremendous impact. First of all, if they infect you or I, and almost every vet student that didn't grow up on a farm gets infected during vet school, then you get prolonged diarrhea, more than 5 days, and it really knocks you out. There are some famous outbreaks when water filtration is broken down. In Milwaukee, there were over 400,000 cases in 1993, and it was said that you couldn't buy toilet paper in a 3-state surrounding region around Wisconsin. It's a horrible problem still in our clinical practice in immunocompromised patients, where people can get fatal Cryptosporidium outbreaks and really can only be treated by trying to reduce their immunosuppressive, because the drugs that we have still lead to relapse over and over and don't act directly enough against the parasite. I certainly had lots of AIDS patients in my clinic back in when I came to the University of Washington in 1986, I guess it was. And if they got cryptosporidiosis, I would tell them, you got to talk to my wife, who's a chaplain, and you got to talk to somebody about your will, because it was often a fatal complication in HIV before there was heart. And then the huge burden that's become really clear from studies I'll show you in a moment is in 6 to 18-month-old children in Africa and Asia in particular, and particularly malnourished children are very susceptible to it. And they're probably partially immunosuppressed from malnutrition. That leads to severe and prolonged diarrhea, and chronic infection in these malnourished children leads to damage to the GI gut, contributing to wasting, stunting, and cognitive impairment. So these kids really don't have a choice. They go through life actually with some permanent changes. And it's also the highest mortality in GI pathogens in this age group. So let's dive into a little bit into the studies that have shown that. The GEM study, or the Global Enteric Multicenter study, was run in Africa and Asia and South America. And there they were looking at kids that had moderate to severe diarrhea, the highest mortality group, and kids that have diarrhea under 5. And the Cryptosporidium was the second most attributable pathogen in moderate severe diarrhea. And it's particularly high in the severe diarrhea in Africa, Asia, and 6 to 18 months old, as I mentioned before. The highest association with death in that study, and particularly in toddlers, so in the first 2 years. And they estimated an annual mortality of about 200,000 deaths. And then the MAL-ED study came along, the malnutrition and enteric disease study. They were following cohorts of patients, malnourished children, over time to see what happened. And with that cohort study, they showed that there was increased frequency of cryptosporidiosis in the people with prolonged and severe diarrhea, but they also showed that it had a significant association with stunting and malnutrition. And many, many other studies have shown impaired cognitive development and stunting following repeated and chronic and even asymptomatic infection with cryptosporidiosis. So the Institute of Health Metrics and Evaluation here at University of Washington estimates that there's 8.2 million disability-adjusted life years, DALYs, essentially a year of healthy life lost every year from cryptosporidiosis, if you include all of the different infection outcomes, and 132,000 deaths. So a huge, huge issue. And a huge issue. And we think that cryptosporidiosis, cryptosporidiosis, I'll show you from this graph, really exceeds all of the other neglected tropical diseases, and yet it's not on the list. So up there, deaths and DALYs, and one at the top These are the big ones like tuberculosis, HIV/AIDS, malaria, et cetera. Those aren't considered neglected tropical diseases. But the WHO keeps a list of neglected tropical diseases topped up there by dengue and others. And you could see dengue has the highest mortality rate. They're going out to about 34,000 children dying a year. But notice cryptosporidiosis in the purple down below goes way beyond that to 132,000 deaths if you take the most conservative estimate. And then in terms of the DALIs, which is the right side of here on the neglected tropical diseases, schistosomiasis leads the list for DALIs. I guess I'm not pointing well. There we go. Schistosomiasis leads the list for DALIs. It's about 3.5 million DALIs, but Cryptosporidium has 8.2 million DALIs. So the bottom line is that really Cryptosporidiosis should be added to the WHO list, and we feel that would help bring up its stature amongst these diseases and help us find interventions for cryptosporidiosis. So we do need new drugs to treat cryptosporidiosis. The current drug that's available is called nitazoxanide. It's the only FDA licensed product. Some people use another drug called paromomycin off-label for it as well. Both of them really only work in the lumen. And nitazoxanide has a number of problems. So first of all, it was licensed with a relatively small signal. There was only 2 days of improvement compared to placebo patients in terms of diarrhea output for the licensure for cryptosporidiosis. So not a huge bettering. It really has marginal efficacy in malnourished children. And in one study in hospitalized patients shown here by Beatrice Amati in Zambia in Lancet in 2002, she was only able to demonstrate a 34% improvement over placebo with this drug. And 3 randomized controlled trials have shown that there's no benefit for HIV-infected patients if they're immunocompromised. And it really makes no sense that it's a good crypto drug because the Cryptosporidium hominis, which causes 70% of the human infections, actually lacks the presumed target of this drug. And for Cryptosporidium parvum, it does have some activity, but only at very high concentrations, and it's unlikely to reach those concentrations in the gut. And we think that it actually, because it works on such a wide variety of pathogens, we think it actually stimulates a gamma interferon response in the gut, and that ends up attenuating the infection somewhat. But there's no, we don't think there's a direct killing effect. And also there's just no activity in immunocompromised hosts or immunosuppressed models. So it's ineffective in the major target populations we really need this drug for, the immunocompromised and malnourished children. So I, together with Wim Hole and Ethan Merritt, a number of years ago, solved the structure of calcium-dependent protein kinase 1 from Cryptosporidium. And calcium-dependent protein kinases are descended from plants into apicoplasts. Apicoplasts are kind of an interesting hodgepodge of plant genes and animal genes. And this was one that came from the plant side. So that was good because that made it easier, in theory, to target it with no mammalian homolog. It's required for replication. That's been shown definitively. And the way it works is when the Cryptosporidium binds to that enteric cell, the enterocyte, there's a flash of calcium that's let out into the cell, and that activates the calcium activating domains shown here in this beautiful colored part of the thing. So that binds 4 calciums, and then what happens is that this colored domain, or the calcium activating domain, moves 180 degrees behind the molecule, and on this side you have the active site. That allows a protein to come in, and a protein kinase wants a protein to dock and then put on a phosphate from an ATP there. So it doesn't work if there's no calcium along, but when it senses a cell, the calcium flash goes off. That allows it to push itself into the cell and then undergo replication. So this was a really cool set of structures that we were able to solve. But more importantly, when Coyote Ojo, who is a postdoc at the time in my lab, looked at the structure carefully, he realized that there was near the adenosine ATP binding site, there was a hydrophobic pocket that wasn't in most mammalian enzymes. So, and this was something that Kavan Shokat had shown at UCSF could be useful. So Kavan had set up where he would mutate whole mice and change a protein kinase so that it would have a small gatekeeper residue like a glycine, and then could use bump kinase inhibitors to target that kinase during life. The kinase inhibitor had no effect on the mice if they were not mutated, so it was a very nice system. But we similarly reasoned this would give us specificity because we could target with these bump kinase inhibitors. We could target the Cryptosporidium CDPK1 very specifically because the typical mammalian protein kinases had this bulky gatekeeper that kept the bump out of this large accessible hydrophobic pocket that was formed by this small gatekeeper, and then we were able to get good inhibition. So it gave us both specificity, so we weren't inhibiting the mammalian kinases, but also great affinity by putting a greasy thing inside of this greasy pocket, if you will. So, so fast forward to, we were funded to do this with Dusty Malley and Erkang Fan, who are in chemistry and biochemistry here at University of Washington. They're medicinal chemists. They made over 800 bump kinase inhibitors that would fit into this thing. And we were guided by at least 87 co-crystallized X-ray structures of CDPK1 with the bump kinase there. So we could tell really carefully what changes we could make to retain potency and what changes we couldn't make to have issues. And we also followed human SARC as another structure to follow along. And SARC has the smallest gatekeeper in the human kinome, so that would allow us to get specificity. If we weren't hitting SARC and we were hitting CDPK1, we were probably getting kinase specificity. Then we set up we call this a snake chart, if you will, but a cascade that we followed for each compound to first in vitro screen them for activity against enzyme, check for mammalian cytotoxicity early on, look for activity against the parasite, then move to PK and toxicity and DMPK studies, which can be done in vitro, some modeling, then an in vitro efficacy study with mice, safety screen that was much more involved, and then finally a large animal challenge with a model and calves that I'll tell you about. So we ended up optimizing potency and efficacy using all these criteria, safety, and then pharmacokinetics and ADME, or absorption, distribution, metabolism, and excretion. Because as I told you, you need to really get into the gut, the whole gut, to expose the compound where the parasite is replicating. So let's see. So again, trying to get the compound with this kind of the right optimal safety, efficacy, and PK/PD was really important. Well, I'll start right off and tell you that it took us about 5 years, but we went through all of the Dusty's compounds, the pyrazolopyrimidine compounds or the PP compounds, and none of them seemed to have all these properties of safety, efficacy, and pharmacokinetics that we needed. So we focused on the aminopyrazole carboxamide compounds, and I'll show you this one that we worked on quite a bit. The bump kinase inhibitor 1770, which was efficacious and safe in many models, but there became an issue with it. So it was efficacious in the mouse model, had great properties for safety, low systemic distribution, long residence in the GI tract, and efficacy in the newborn calf model of C. parvum. But then this thing emerged in calves that we were treating with BKI 1770. They underwent a strange stance, and they preferred to kind of lie down. I'll show you a picture of it in a moment. So this was like the good old days when I used to play these board games like Chutes and Ladders. How many of you have played Chutes and Ladders? Yeah. Oh, good. Quite a number of you. So you're probably experienced enough to know when you get close to the top, if you roll into a chute, you go all the way down to the bottom again if you hit the wrong chute. And this was the wrong chute. So we basically had to abandon this compound. During that time, we were working together with an animal health group, and they had treated some animals in a safety study and noticed this issue with dropped pasterns and the plantigrade stance. So the plantigrade stance is, notice they're sort of on point, almost like a ballerina here. It's a little hard to see there. And here, their ankles are more or less dropped. And also their toes are splayed out, right? Their hooves. And this is typical of the plantigrade stance and the drop pasterns. And they were also noted to be somewhat reluctant to arise. In looking at the bones of the animals that were treated here, we could see that there was chondrocyte hypertrophy. So you can see here the chondrocytes in the treated, but not in the controls, have greatly hypertrophied. There was swelling below in the vascular zone down here, and the epiphysis, which is this purple area, was uniformly thickened in these. So there basically was no safety margin for this drug, and we had to abandon it. But it didn't correlate with anything we were testing in vivo, in vitro, or any observations we made up to this point. So it was a bit upsetting, as you can imagine. But Ryan Choi and Matt Halverson, two wonderful research scientists in my lab, got together and developed this mouse model for bone toxicity, and we were able to reproduce what was happening in the calves in terms of the bone toxicity. So you could see this compound here, 1770, if they treated the animals for 7 days with this relatively high dose of the bump kinase inhibitor, and then sacrificed them and did bone histology and just measured the tibial epiphyseal plate, and they were blinded when they measured it, compared both the left and the right tibia, they could find a significant difference for this compound versus the ones that were treated on the control. And in fact, they found another compound that hadn't progressed as far, gave the same phenotype, this BKI 1745. But these two compounds, BKI 1708 and BKI 1841, appeared free from this kind of bone toxicity. So we were happy about that. We actually pursued the BKI 1841 because we knew it already. It had a lot of safety properties that we liked. It was efficacious in the mouse model. There was no genotox or developmental toxicity signals that would prevent young children and pregnant women from getting it. It was safe to mice given at high dose. No bone toxicity, I just showed you. And then we did this C. parvum newborn calf model, and we were very happy that it appeared to be very efficacious. In fact, Diarrhea stopped after 1 day of therapy, and there was good parasite efficacy even with 2 mg per kg given twice a day for 5 days, a relatively low dose. But then oops, got the wrong thing. Sorry about that. Then calves developed this same sort of posture with the drop pasterns, the plantigrade stance, and so on the last day of treatment. It was fully reversible by day 10, but that confused us because we had this hypothesis that this was from the bone toxicity, and yet this compound didn't seem to have it in the mouse model. We then talked to our tox advisors, and they said, this smells more like CNS toxicity, central nervous system toxicity. They were helping us actually from AbbVie Global Health that was helping develop drugs, and they were helping us a great. So again, down the chutes and ladders with this compound wasn't something you'd want to put in young children, but the group at AbbVie had this up and going CNS toxicity model. So the idea is you dose animals, and in this case mice, repeatedly, and on the final dose, after 5 days of dosing, you put them in a box with lasers that are going around and basically measure how much the mice move around and so on, and compare treated and gavage control animals. And what you can see here, it's a rather complex thing, but this part of it is distance traveled, and this part of it is rearing vertical activity. And you can see in the controls, which are blue, that the rearing activity, even at 20 minutes after the dose, is significantly lower for the 2 compounds that had the problems, 1841 and 1770. And again, here at later time points, they're rearing up less, which means they're probably CNS depressed. And that also correlated with distance traveled, which came out significantly more a little bit later. But we didn't see that in this drug, BKI 1708. So it really suggested that the BKI 7070 had both the bone tox and the CNS problem, but 1841 had the CNS toxicity. BKI 1708 appeared to lack these issues, so we moved on with that. So I'd like to take you through that. That's our preclinical candidate at this point, and it appears really hopeful that it'll go all the way and fit just the kind of drug we need for this thing. So again, this was made in Erkang Fan's lab, and he championed these amino carboxamide drugs, the AC drugs. It had picomolar activity against the enzyme in vitro, and great activity against the parasite shown here at 410 nanomolar. No activity against mammalian cells over to the right of that. The HERG has a slight yellow warning thing. It's 12.3 micromolar, but that's the free drug in circulation. You can see here that it's highly plasma protein bound, so that should protect it from the negative cardiac effects of blocking HERG. No mutagenesis or genotox signal. We put this in mice at very high concentrations, and we're unable to see any abnormalities there as well, and had good PK activity. So this is our mouse model that I've been mentioning but haven't shown yet, and this is N-luciferase tagged Cryptosporidium parvum parasite. Boris Streepen's lab made this, and we use it because it's really easy to quantitate the output of the Cryptosporidium oocysts. That we're not left there with countless hours of microscopy. We just dilute the stool into luciferase substrate and measure the relative light units, and that turns out to be directly correlated to however you measure the Cryptosporidium oocyst output. And so if you don't treat the mice, they generally get very high. So in the red here, where's my pointer? There. Well, in the red bar, you can see the untreated, and you can see how it stops in the middle of the infection, and that's because when mice, they don't get diarrhea, but if they lose weight beyond 20% of their body weight, then we have to euthanize them. So often the controls need to stop. And then you can see a dose response here of the 1708. It was given daily times 3 days. So treated here at days 6, 7, 8, and 9 after infection, after the infection's very well established. And you can see down to 15 mg per kg, shown here in the purple line, the parasites are eventually eliminated below the level of detection, the red dotted line above. 8 mg per kg, as you can see, was not quite effective. So we found excellent parasitologic outcomes with this BKI just given for 3 days at a relatively low dose. The in vitro efficacy, ADME, and toxicology was excellent. And there was importantly, there was no signal in advanced cardiac testing, central nervous system, bone, or developmental tox. One thing that emerged early in this program with this compound is we realized that there were a number of metabolites made, but one in particular, this M2 metabolite, seemed to predominate in vivo in all the models we looked at, particularly in mouse models. And that was due to dehydrogenation. And then keep thinking you're seeing what I'm pointing at there. Dehydrogenation, and then it isomerizes into this hemiaminol, or this extra ring that's formed. And this is quite stable, and in certain species it becomes the predominant driver. And that turns out actually to be an active metabolite. The other metabolites really aren't active and don't hang around very much, but this one does. So it's interesting, when it's converted to this hemiaminol, it retains activity against C. parvum, almost as active as the original compound. You can see above it there, so 0.73 micromolar versus 0.41. But what's also interesting, particularly in the mice, this is the parent compound, the BKI 1708. This metabolite builds up to higher levels in the plasma than the parent ever does. And it retains, it also is retained highly in the distal gut, like the ileum and the colon, probably driving most of the activity. And in fact, we dosed animals similar to that thing I was showing you with just the metabolite. It's difficult to make, but Erkang Fang's group made enough of it for us to test. And we could see great activity down to 15 mg per kg. So suggesting that most of the activity that we were getting out of the BKI 1708 was actually due to this, and it had good pharmacokinetics in terms of distribution through the bowel for treatment of Cryptosporidium. So here's a shot of one of our solved structures. Scott Lovell at University of Kansas did this for us very recently. Here's BKI 1708. Here's the bump sitting in that hydrophobic pocket I was telling you that gives us this specificity for the bump kinase inhibitor. And here's the BKI 1708 making the 3 canonical hydrogen bonds that bind adenosine or adenosine triphosphate well into the ATP binding pocket, stabilizing the molecule. And what's interesting perhaps is that the metabolite 2, or 1862, here with the neocyclized part, only makes 2 of those 3 hydrogen bonds. And we think that's the reason that the enzyme activity is largely lost by this compound. So that gives us a quandary about what it actually is working on, because we've only gone down in activity by about 60% or so, but yet we've lost 100-fold on the target we thought we were hitting here. So that suggests to us that actually this is hitting a different target, and perhaps these two, the metabolite and the parent, provide dual targeting, which actually would be a great thing for emergence of resistance, which is a problem in Plasmodium for sure in this class of organisms. So that's actually attractive to us for this compound. So one final data slide showing you the newborn calf model. So this was run by Mike Riggs and Debbie Schaefer at University of Arizona for us. And what they do was they would collect newborn calves and catch them before they would actually get infected by Cryptosporidium in the farm they were being born in, bring them into their facility, let them recover for a few days, and then infect them with Cryptosporidiosis. And then once they were just initiating diarrhea, they would start to either treat or give them mock treatment. This is showing treatment with our compound. Here in the light gray is the BKI treatment with BKI 1708, and at 2 different doses, a low dose shown in the light green and a high dose shown in the dark green. And you can see that the peristologic effect was much better with the higher dose than the low dose in the number of oocyst counts that are shown here. Interestingly, the clinical evaluation score, which is how the calves look to blinded vets, gets a lot better in both groups very quickly. Fecal volume, which is a measure of the total volume put out by the calf over the day, shows a really quick response, basically down to normal by the end of treatment. And fecal consistency with stools being very watery up here and very well formed down here gets quickly with both doses. So our conclusion was there really was excellent clinical response with both of these, but the peristologic response looks much better with the higher dose. And then one other thing was that Debbie Schaefer took the time to isolate the parasites from the days that were treated and then the days afterwards that they were collecting in 2 groups and tested them for infectivity. And basically, the early collected parasites infected almost as well. This is reduction in infection, so only a 20% reduction in infection for early parasites, but later collections after the treatment was over showed over an 85% reduction in infectivity. So there were small amounts of parasites that were released into the environment here, but they're mostly dead. And so we feel that this is going to have a better public health effect, both in many logs reduction in terms of oocyst output, but also many of the oocysts that can be obtained are actually functionally dead. Okay, so what's our next steps in the development of this? We've completed 14-day studies in 2 rodent species, and we didn't find any tox looking at histology, biochemicals, and everything, even getting over 20-fold more than efficacious exposure. We completed a similar dose-finding study in dogs and found no dose-limiting toxicity. So we're moving on to, and we've completed good manufacturing practices studies with showing that we can demonstrate a 2-step synthesis at very low cost of goods, so really hopeful for a neglected disease drug. We can get over 99% purity just with recrystallization. And we've shown that we can make a pediatric drug formulation with just 2 low-cost excipients and mix it with water for our clinical trials. So we're very happy about that. So the next steps are expensive steps, which a recent grant from CPATH has helped me with. They have a translation accelerator grant, and they've given us $500,000 to complete the GMP manufacturing and to do the good laboratory practice rat and dog studies that'll be carried out at a contract research organization. And they're underway right now. So we hope to be filing our IND in November We've already had a pre-IND meeting with the FDA, and they've told us that we're on track with the studies we're looking at here, and very excited about eventually moving into clinical trials. Do I have time for one or two more slides? Okay. I just want to talk to you about, okay, you may be sitting there thinking, well, what the heck is the pathway? You know, most of the people you can find that are infected with cryptosporidiosis in the world, Cryptosporidium, are in Africa and Asia, and they're 6 months old, 18 months old. So this is a pathway we're somewhat piggybacking on what malaria does, but we've gotten through most of the IND enabling steps. And then meanwhile, another group is working on a Cryptosporidium drug at Novartis, and they've already developed a controlled human infection model, or CHIM, for Cryptosporidium. So they're dosing adults with Cryptosporidium that are GLP prepared and blessed by the FDA for whatever that's worth. And then people are willing to do that and take a placebo or drug to see if that'll actually treat their thing. So our plan is to get a first-in-human study done in healthy volunteers and then move into phase 2A controlled human infection trial. We've mapped out how that would happen. Then there's some relatively long and expensive juvenile tox studies that need to be done before you move into a pediatric use, but that's our plan. And then go directly to the target population that you can find every day in Africa and Asia. That is the population that's suffering from cryptosporidiosis. And I don't think there's enough time to talk about the use cases and so on, but if you have questions about that, we can talk about that at the end. But just to summarize this, we talked today about the need for cryptosporidiosis therapy, that BKIs, or bump kinase inhibitors, look very good. They're potent in vitro and vivo. They give us specificity. They target this calcium-dependent protein kinase 1. They had off-target safety issues, which we actually could never molecularly explain, but we made models to figure out what we could mimic them with, and then drove them out of the molecules that we studied. And our current lead looks really great. It might have the additional blessing, if you will, of making 2 compounds for 1 in the human body, like we're predicting, and may provide dual targeting. We're studying that to try and figure out what the other target might be in Cryptosporidium. And the steps to IND, or Investigative New Drug, are ongoing and expected by the end of the year, and the pathway for clinical trials is ahead of us. So developing drugs is a team sport. There's a huge group of people that helped us. I want to particularly call out the scientists of AbbVie that did a lot of safety studies for me, the research scientists that have been with me from the beginning, Matt Hulverson and Ryan Choi, and Lynn Barrett, the lab manager, has been with me for 37 years. Pretty amazing. But all of these people helped at one point. The funding came from a lot of sources, including PATH, Gates, USDA, and NIH. And if you're interested in cryptosporidiosis, join us on LinkedIn for the Neglected Parasite Initiative, or join us in the Cryptosporidiosis Therapeutics Advocacy Group. And thank you very much for your attention. I see some questions.
Thanks. That was a tour de force, really, of chemistry and biology. I have a related question that's not so chemistry related. I think a lot of people know it's not apocryphal, real story when someone put rabbits into Australia and then tried to get rid of all rabbits by bringing in boxes of measles, and that was a problem, didn't work. But what a lot of people don't know is that eventually they killed the rabbits with viruses. Do Cryptosporidia have parasites themselves or pathogens that will, because I know people that tried to treat and have successfully treated bacteriophage, and I'm wondering if that could be a complementary approach to, small molecules for eradication to have something that's persistent and try to outcompete it or kill it. Mammoth.
That's such a great idea. And I'm not aware of viruses for cryptosporidiosis or other pathogens. There certainly are and have been looked at in trypanosomatids, other protozoan pathogens, and they've been looked at for perhaps co-treating with drug trypanosomatids such as Trypanosoma cruzi, Leishmania, and so on. But I'm not aware of it in the Cryptosporidium group. Yeah. Yes.
Let's have a couple of questions. I'll start with just one. It sounded like early in your talk, you hinted that if you survive an immunocompetent person who survives and clears an initial infection is then immune. If that's true, what is then the potential impact of sort of treatment? Are people going to be susceptible to reinfection and sort of related to the data you presented on oocyte?
Yeah. That's really interesting because I think, tell me if I'm not answering this question because I'm famous for that, but in the AIDS era, we would treat people with nitazoxanide and paramomycin, and they would get an amazing response and start to look healthy again. We'd have to stop the drugs and it would come roaring back. And on autopsy, they found that the biliary tract was just full of the organism and that the compounds really weren't making it up into the biliary tract and providing clearance there. Since that time, they've also realized that there's a pulmonary form of cryptosporidiosis that probably persists. So I mentioned Novartis is developing a cryptosporidiosis drug, but theirs is what's called a soft drug that won't get any systemic distribution. Ours will get decent systemic distribution, we're predicting in humans, and should take care of the biliary tract problem and perhaps the pulmonary problem as well. But is that what you were asking, or did I answer it wholly?
Interesting. I have to say, I think it's one piece.
Yes.
You don't need very many oocysts to survive.
Yes. Yeah, you could get infected by your environment if you've got oocysts all over your home now. You're right. Yeah. Is that what you meant? Yeah.
Interesting composition. Thank you.
Brad?
Thanks, Wes. That was really great. You mentioned that there's the active metabolite of 1708 that you don't know what the target is, but can you make that active metabolite, and is it active by itself?
Yes. So yes and yes. We've actually been able to make. It's actually very, very difficult to make for Urkang-fan, and we've been trying to come up with another synthesis because one possible response the FDA may have is, well, just make the metabolite and use that and don't bother with the other. But it's actually very difficult to synthesize and relatively unstable. This one, we actually have been able to, we've had it stored at room temperature for 3 years, the BKI 1708, and it's rock stable at room temperature for 3 years. Even heating it and putting it in a human environment. But I think your question's a good one. We were able to synthesize enough. We fed it to infected mice and showed that they would clear just as if we had given them the BKI alone. It's interesting. The thing is not quite as extreme in all the other species we've looked at. So and we've looked at rats, monkeys, dogs, cows, horses, for various other reasons. And generally the metabolite level comes up only to about 30% of that of the parent. So mice are really an outlier. We're trying now to find out exactly how the metabolite is made. And people from the Department of Pharmaceutics are helping us, Dr. Toda over there. And then we'll be able to better predict whether humans are going to have exposure more like mice or more like all the other species we've looked at. Yes.
Wes, good to see you.
Good to see you too. Thanks for coming.
Sure. So I was wondering, given the current FDA and federal government interest in infectious disease, or perhaps and tick leak disease that might affect people in lower income countries. Is it going to be harder now to try to get these kind of stuff, get this drug out for actually helping people?
In theory, although I think we can play it both ways, that as people here from the microgroup know, we see, especially in our immunocompromised, that Americans are quite susceptible to this disease too. And what we have now is really not helping. So this is a really great orphan drug for Make America Healthy Again, if you will. Yeah.
About other coccidian parasites, is this like a path to addressing some of the lesser known ones as well, or do we need that?
Yes. Yeah. So the question was, what about other coccidian parasites? I mentioned, and you may have seen up there, we have activity against Toxoplasma, and we're looking at this actually for Toxoplasma. In the mouse model of Toxoplasma, it works really well at clearing oocysts from both the brain. And if you take, so you guys probably know that Toxoplasma is the scariest thing if you're in your, pregnant and you get Toxoplasma for the first time. There's a mouse model for that, and when we treat them with this, they completely clears them and their fetuses. So we're very excited about this as a drug for toxoplasmosis too. There's a sheep model for that as well, and we've looked at a slight different one we used in the sheep model because this one had horrible pharmacokinetics in sheep, and it worked beautifully in terms of saving sheep from loss of fetuses as well. Interestingly, it doesn't hit malaria. The small gatekeeper calcium-dependent protein kinase in malaria is the protein kinase that's important for exflagellation. So it stops the sexual cycle. So we were thinking if we could get this up to certain blood levels in humans and had zero toxicity, it might be transmission blocking in areas. So Coyote Ojo in my lab is following up on that. And then there's a variety of coccidian animal diseases that this has some benefits for as well. So. Thank you. Thank you all for coming.